Hello, everyone, and welcome to today's webinar, Eavesdropping on Biological Processes with Multimodal and Multiplex Imaging. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more, visit them at leica-microsystems.com. Now, we encourage you to participate today by submitting any questions you may have during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of this presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. I'd now like to welcome our speaker, Dr. Scott Fraser, from the Elizabeth Garrett Chair in Convergent Biosciences. He's the Provost and Professor of Biology and Biomedical Engineering and the Director of Science Initiatives at University of Southern California. Scott, welcome. You may now begin your presentation, sir.
Well, thank you very much for that kind introduction and for having me here today to tell you about our work using imaging to watch biological processes as they're taking place in systems as diverse as I'm showing here. A lymph node where we're watching cells from the outside coming into the lymph node and communicating, and a beating heart on the far right. I've been very active in trying to develop interdisciplinary tools and in translating them out into the marketplace, so I have a few disclosures to make. And what I'm going to try to tell you about today is the work that we've been doing working on embryos as a system that allows us to do analyses of cells in their normal setting. Why we like embryos is that they exercise a good fraction of their genome quickly enough in a small enough package that we can use imaging to an advantage. Now, many of the studies right now, and most of the systems analysis studies, involve single cell techniques. And these single cell techniques have been incredibly powerful for discovering proteins and discovering the genes that code for those proteins, but they haven't been as good as keeping the cells in their normal setting and capturing what the dynamics are of the cells interacting with one another. They have shown a great bit of heterogeneity and variability, and that's often a complication. What we're trying to do is to use imaging to move those systems analyses into embodied cells, as we like to say. This way, we are looking at the cells in their normal setting, and the dynamics and the variance isn't something for us to curse at. Instead, that dynamics and that variance is the signal that we're using to understand the underlying processes. But of course, that requires the sort of multiplex and multimodal tools that I'm going to talk about today. I'm going to start by just mentioning the tools we've been using for looking in fixed preparations. These fixed preparations, we would like to be able to understand where genes are expressed. And so to be able to do that in a quantitative fashion and being able to look at multiple target RNAs, we've gone over to an approach called hybridization chain reaction, developed together with our colleagues at Caltech at the time, Niles Pierce and his laboratory. This technology depends on hairpins that are happy in solution interacting with themselves, as you can see in this simple cartoon of a hairpin. Even though they would like to bind to one another, they're trapped in that state by all the base pairing across the stem. And so they are trapped in that metastable state until they see an initiator sequence. That initiator sequence, I've got A star here marked, is complementary to the A of one of the toe holds. And when it binds, now it can open up this hairpin by branch migration because it has a sequence that will complement the hairpin sequence and open it up. That exposes C star B star, which happens to be the initiator for the other hairpin. And so this goes on, regenerating the initial initiator and growing explosively. Because this relies on sequence identity between the initiator and the hairpin, this can be done in a multiplex fashion. And so if we take 4 different hairpin pairs labeled with distinct dyes, we can see that these hairpins on a gel will run to the small molecular low domain here in the gel in the presence of just blender extract of tissue or any other sort of non-specific sequences being added. But if we add initiator for, say, hairpin pair 1, the blue appears as a 200 mer or so of hairpins and disappears from the small hairpins that are individual. Instead, if I add initiator 3, the green appears and disappears from below. Now, this can be turned into a way to look at the distribution of RNAs in a tissue by using antisense probes to recognize the RNA, then initiator sequences flanking those. And when hairpins are added, then they self-assemble. And each place that there's an initiator, we get about 200 dyes being deposited. That's enough for us to see deep within tissue, and the reagents are small enough to penetrate well. So it gives us a very reliable gain that's very linear, and it allows us to see fine features. Unlike, for example, in the right, you can see a conventional in situ hybridization. You can see the conventional in situ hybridization, in this case, looked at with fluorescence. And if we look instead at the HCR, you can see that even the nascent transcripts within the nucleus, these dots, show up clearly, as well as the localization of RNA within the cell. Because it's got such good capture efficiency and such good linearity, we can even detect the same message in more than one color. And this allows us to validate both the efficiency and the linearity of the gain. In this case, I'm detecting the same RNA with red and green hairpins. You can see the 2 images here. And if we plot it on a pixel by pixel basis, you can see that they are clearly greater than the background signal. And the nice clustered linear plot shows us that we've got good linear gain and good discrimination ability, signal from noise. This approach, of course, brings up one of the problems that we have whenever we're using imaging. And that problem is that we're always trying to compromise between opposed goals. We would like to be able to image at infinite resolution, infinitely quickly, and throughout the depth of a specimen. And we're often frustrated by the fact that as we try to go to higher resolution, that typically sacrifices speed and depth and so on. And lurking in the background is the enabling photon budget. How many photons are there being given off per pixel per unit time? To try to make this frustration less frustrating, we've gone over to a slightly improved version, which we're calling Muse, that allows us to conveniently get a larger gain and also to use distinct labels for different imaging modalities. Just like before, this relies on an initiator sequence, and here I've cartooned it differently as these chevrons. The chevrons give us the same sequence-specific orthogonal gain, and we can then later put on dyes or metals that allow us to see this within a CyTOF instrument. And since this initiator can go onto proteins like ligands that bind to other proteins or antibodies, or onto RNAs, we can easily go into tissue and see the protein and see the RNA. We're excited about it because it allows us to use labels that are inconvenient in most preparations, like quantum dots, and pick up both the spectral advantages of quantum dots or other sort of dot-like structures that can be a couple of orders of magnitude more photon budget. It also gives us a greater gain. You can see that if we run just our standard muse or our higher gain muse, we can pick up 1 or 2 orders of magnitude in intensity. And we can also see proteins. And in this case, we're looking at fly embryos, which we find to be a very convenient system to test our developments. Because we can put these initiators on antibodies, and those antibodies can be directly labeled, for example, rabbit primary antibodies, we can do 4-plex recognition using no secondary antibodies and using antibodies all from the same species, in this case, all primary antibodies from rabbits. We can look at RNAs, and you can see the individual dots. And if I was to blow these up, you would see that they are nicely orthogonal. None of the dots are double labeled. And we can also, of course, do simultaneous immunofluorescence or other ligand binding studies with the RNA studies. So this technique has really allowed us to go into settings and ask where genes are expressed, and allowed us to test the results of single cell studies that have been performed looking at the proteome or the transcriptome. While it allows us to see those and allows us to see many of them in context, the bad news is that there's 5 gigavoxels worth of imaging data here. And in a conventional confocal microscope, that can be a significant amount of time. And we typically have to do tiled acquisition. So this is 4 by 6 image stacks. And you can see the slight bit of bleaching at the borders because it was imaged twice. So we are, of course, wanting to get rid of this bad news. I'll tell you a little bit about that. The other bad news is we'd like to be doing more than 3 or 4 colors, and I'll spend the balance of my time talking about that. So because biological imaging has no free lunch, we always live in this pyramid of frustration. One thing we're wondering is about the, sort of minimizing the cost of that lunch. And one way to minimize the cost of that lunch is to move from point scanning microscopy, a confocal or a 2-photon microscope, to light sheet microscopy, where we bring in a light sheet from the side to do the excitation of the fluorochrome and collect the image in parallel. This allows us to benefit from the Moore's law sort of development in the capabilities of cameras that can acquire a low noise image very rapidly of a whole image plane. To get deeper imaging and at lower phototoxicity, we've gone over from the normal blue light excitation, as you're seeing here, which over here to the right, you can see a laser beam moving across the side. You can see in this slightly scattering media, the labeled beads in this case within gel showing up. But you can see that there's a finite width to this light sheet, and it tapers to a minimum thickness at its focus. But any light scattering can give us some excitation outside of the focal plane. 2-photon requires on the simultaneous absorption of 2 infrared photons. Because of that, we get no excitation when the light's not focused down. In that central focal region, you can see we get a very nice, clean light sheet excitation domain with a distinct start and stop. And this allows us to go deeper into the preparation without seeing blurring from scattering. And it also allows us to see very dynamic things without any sign of phototoxicity. This is a fly embryo again, watching it develop on the stage of the microscope. And fly embryos developed on the stage of the microscope, but not in the beam or just out on the lab bench, or at exactly the same pace, and eventually hatch the same way. Because it's infrared light, It also allows us to go deep into structures that might otherwise respond to light. In this case, we're looking at the calcium signal from a light sheet recording of a zebrafish. You can see him here in being held within the field of view of the microscope. And what I hope you can see is the individual bright spots as there's activity in the brain, using calcium level as the surrogate for looking at the neuronal activity. And we can see throughout the depth of the brain, this is part of a 2-day-long time-lapse movie without appreciable photo bleaching, which really shows the power of 2-photon light sheet. That 2-photon light sheet lets us move from hours to minutes for our imaging, lets us capture dynamic structures like this beating embryonic heart that you're seeing to the right, and even lets us see at subcellular resolution. For example, you can see the individual cells of the cardiomyocytes here as the heart is beating. So back to our question of no free lunch. I've just told you about ways that we can increase the photon budget. I've told you about ways that we can increase the speed and depth by going to 2-photon light sheet. What about increasing the multiplexing? I want to look at many different labels at one time. And if I want to look at those many labels at one time, I need some way of carrying more colors into the situation. And I'd like to do it with a real-time sort of processing. And this problem, anything having to do with fluorochromes and trying to look at more colors, is always plagued by the problem of looking at a spectrum that is finite in its width. In fact, it's wider than the difference between a yellow fluorescent protein and a green fluorescent protein. So the cartoon above shows what we hope to see. If we look at it in true color, the blood vessels and the muscle cells are not that distinguishable by eye. On a normal confocal microscope, or a light sheet microscope, where we've got multiple fluorescent proteins like blue fluorescent protein, green, yellow, and some of the red fluorescent proteins, and of course some of the background autofluorescence, you can see what the challenge is because many of these spectra overlap considerably. And our temptation is always to use narrower and narrower, more selective filters to try to see that signal. But as you can see, even if I make a narrow detection filter, there's still a significant amount of yellow fluorescent protein coming through in the green fluorescent protein signal. And even if I went down to a very, very small window, I'd still have other things bleeding through. And of course, I'd be throwing away most of my photons. And so I'd be hurting myself on the pyramid of frustration. Years ago, we began trying to deal with this instead of with narrower bandpass filters, using approaches that were borrowed from the remote sensing crowd, the people that fly satellites over the Earth, for example, in which they collect a spectral cube that's like an image stack, but it's a spectral stack or a lambda stack. So X, Y, and wavelength. We built microscopes that could see multiple wavelengths, for example, by using a grating and a multi-window PMT. And even commercial units have been able to execute this. This showing the Zeiss unit for doing multispectral detection. The approach is very simple. It normally uses an approach called linear unmixing, where knowing the basis spectra and a number of spectral bands collected at once, you can see that GFP and fluorescein overlap in every one of these spectral bands. But knowing what this basis spectrum is, it's possible to unmix the two. Now, this has the advantage that it lets us see dyes that are very much overlapping in their spectra as distinct, but it involves a significant processing time. And it also involves the problem that the noise of the least bright label gets imposed on the brighter labels. And you might be able to see the photon counting noise of the green label being imposed on the red label in this unmixing. Luckily, an approach from Enrico Grattan and his colleagues of using the Fraser, a Fourier transform of the spectrum, gives us a way to speed the processing and improve the performance. In this approach, a region that's red here and would have this red spectrum, when we take the Fourier transform and plot the real and imaginary terms of one of the harmonics of that Fourier transform in what he calls the Fraser plot, we've got here a red dot for that red spectrum. The green spectrum becomes a green dot, and a 50-50 mix, instead of being this double humped thing that we have to try to do linear unmixing on, becomes a dot halfway in between. A 70-30 mix or a 30-70 mix is positioned in between the two, just as you'd guess from the ratios. This has allowed us to do hyperspectral Fraser on the same imaging data, trying to separate it out optically, separating it out by linear unmixing, where you can see the photon counting noise being imposed in the other channels, and where the 5 gigavoxels of data can take some time to process it in many settings, and the hyperspectral Fraser, which can be far less than a second of processing, in fact, more on the millisecond scale. This allows us to cleanly see the blood vessels and the muscle cells as two separate structures. And it lets us look, for example, in this animation, at several different fluorescent proteins in the same living specimen. So even though the spectra overlap considerably, we can separate the signals nicely and even collect the signals using non-ideal excitation wavelengths. For example, we use two different excitation lasers in this case, one at 458, which is not what you'd normally choose for GFP or citrine, but it allows us to use the unmixing very actively to get our different signals. We've been using a combination, or what we call hybrid unmixing, as a way to capture that speed and that ability to do multiplexing. And this cartoon helps us understand why it works. Our normal hyperspectral data set of something like a zebrafish embryo would involve us doing linear unmixing on each of the pixels, or each of the voxels, within this specimen, which can be tens of millions, if not more, different things. And so that takes some time. If we instead reduce this into the hyperspectral Fraser world and do some spectral denoising, we can change, or we can process the spectra without degrading the spatial resolution. We can bin this on the spectral Fraser diagram and then do the unmixing now on each one of these pixels, and use that to try to do the same sort of unmixing, but now on far fewer pixels, so we can work far, far faster and get much cleaner, lower noise results. Now, if we want to carry that to an extreme, and that accounts, by the way, for the very fast processing time that we can get good performance. But now what we really want to do is go in that pyramid of frustration towards the lowest number of photons being collected, the fewest excitation events. And as we do that, these nice spectra become noisy because of the photon counting that happens as we have fewer and fewer photons. We have used machine learning to try to reconstruct the spectra using the noisy photon-starved spectra and the known spectra to try to predict what was there and see if we can get an approach that lets us reconstruct the spectra at minimal light doses. This means we have to do a huge training set, and while we could do this by collecting ground truth images and the low signal images, we can also do it by building and validating a very good model of the microscope and using that to generate spectral data sets at low signal and at low noise, and training the machine learning algorithm. This approach has allowed us to generate very useful data, for example, of cell boundaries and structures in the perinuclear space, which are largely obscured as we go to low noise imaging, or low photon imaging, by the noise that's there. And it allows us now to collect images with far fewer photons than ever before, and with far greater dynamic range than ever before. So we can look at the signal in the presence of background noise and autofluorescence, and in that same image, even separate out the different contributions from cell types that autofluoresce, like the xanthophores, labels that are within the cells, like the retinoids, or the metabolic signatures like NADH free and bound that we can use to try to see the metabolism of the individual cells. So this hybrid and mixing approach has allowed us to go in and see things that normally we would be just invisible from our site because the specimen is so light sensitive. Now, we're very interested in applying this to settings that are not like this zebrafish, where we've got multiple fluorescent proteins. For example, in patient specimens, very few patient specimens come in with GFP and other fluorescent protein labels. And what you can see is, using the spectral signal, we can separate out metabolic signals, intrinsic labels that are of biological signaling importance, and of course other metabolic sort of signatures that are around. But to try to get these signals, what we're trying to do now is to go away from just spectral signals and see if we can move to lifetime imaging to get a far more reliable way of seeing things. This approach relies not on looking at the wavelength of the emitted light, but the time delay between the excitation and the emission, which would normally be an exponential. And we can do this at relatively low photon counts by collecting multiple periods and counting the individual photons, reconstructing that exponential after a laser pulse. The power of this is that you can unmix the different components, and in this multi-exponential decay that's happening, identify the different components, just as we are doing with the linear unmixing. That is doable, but painful. And luckily, Enrico Gratton developed his Fraser approach for looking at lifetime data, in which it allows us, again, to plot the lifetime of things by taking the Fourier transform and to unmix the amount of different components by looking at the geometry of the signal in between two. So this approach that's been developed and really formalized at UC Irvine within the Laboratory for Fluorescence Dynamics really offers us a way to take those multi-exponentials and put them onto this universal circle, in which we can see very short to very long lifetimes, and any combination of the multiple labels that are there will be found at some position in between them. For example, if we look at cell metabolism, free NADH versus bound NADH, that is free in solution in the cell, which happens to be the way that cells are when they're in glycolysis. NADH is bound to enzyme in cells that are undergoing oxidative phosphorylation, so they have slower lifetimes. And you can see that those, depending on the enzyme, get moved over here to the left. So that means that there's a convenient way to follow, using the Fraser and using the unmixing that we can do on the Fraser plot, the background autofluorescence from the metabolic signal, and see something as simple as cells being fed here, going from one metabolic state to another when fresh medium is added to the plate. It also means that we can use metabolism to look into unlabeled structures. And this, again, is from the Enrico Gratton and colleagues' work, looking at intact intestines and looking for the metabolic signature that's present in the stem cells within that. What they were able to show is that stem cells and different transient progenitors have different lifetimes, different positions on this, this Fraser plot that allows them to cluster them. Now, all of this is doable, and we've had equipment that we've built at home or purchased that can do this sort of imaging, but it has not been easy, and it's been a challenge to train users. This got much easier recently with Leica embracing lifetime imaging, and we've been able now to train clinicians, clinical scientists, undergrads, high school students in doing metabolic imaging now. And this just shows one image or one set of images collected with the Leica instrument that makes it far easier to analyze. Here using Fraser to follow as the yellow signature that's present in the side population, or some people might even call them cancer stem cell state, as it's lost over time, the same cell becoming a non-side population or non-stem cell state over the course of a day. This technology has allowed us to look at systems we've never thought we'd be able to before, and we've been able to collect images and actually, well, train users and have users train other users. So it's been quite an improvement. So what about in spectral Fraser imaging? Can we get a similar advance there? And just recently, there's been a similar effort in making friendly approaches for doing multispectral imaging. You can see here a rendering, a sort of cutaway view of a multispectral detector inside of the new Leica Mica. We have been using and training users on this microscope now. And what we like about it is within a single exposure, it can collect images in wide field or in confocal mode. Because the specimen is held inside of this dark enclosure, it's possible for us to sit this in the lab with its support electronics and its incubation and everything else underneath, so that we can collect these images in a normal setting rather than in a dark room. And again, this is a technique that's allowed, or a tool, I should say, that's allowing people to train one another in this powerful technique of hyperspectral Fraser imaging. So I'm going to end there and talk and just sort of summarize quickly how we've tried to add dimensions to imaging. I've told you a little bit about multiplex imaging of RNAs and our approach to try to add dimensions by adding spectral Fraser analysis or FLIM analysis. I hope I've given you a little taste of the idea of using these approaches to try to look into specimens that are very challenging. And I'll just sort of end with two teasers that are shown here in the lower right. You can see a beating heart where we're looking at both the blood cells inside and the myocardium and following them in 3D, and watching an active brain, in this case of a zebrafish larva, as it's responding to visual cues. What we're hoping is that these approaches with these new, more friendly tools are going to let us really go after the things that we thought were impossible, or at least impractical, like watching memories form within an intact animal. And I'm going to end by thanking the teams of people that have made each of these tools possible. The two that I just hinted about are shown over here to the right, but the people that really made each of these tools work and sing are shown here to the left side of the slide. And of course, we owe a huge debt to them and to the funding agencies that have made this all possible. So thank you for your attention, and I'd be really happy to take any questions that you might have on this work.
And thank you, Scott, for that informative presentation. We will now start the live Q&A portion of this webinar. If you have any questions you want to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's get started. Looks like we already have some great questions coming in, Scott. Our first question, you say lifetime is underused. Why has it been so underused?
Well, lifetime requires special instrumentation to be able to acquire it, but it also requires people to think about the way they acquire the images. They have to be able to not just see what wavelength of spectrum of the emitted fluorochrome is, but how long after the pulse of a laser light, for example, the emission is taking place. This equipment has not always been very user-friendly, and it's also equipment that is less commonly available. Why we're excited about it is now that we have equipment that's more user-friendly, we're finding that the users are recruiting other users, and in fact, the users are training other users in collecting the data. And that's sort of a newfound thing for us. I think I might have mentioned that during the talk, that previously the work was doable but painful, and it's now quite a bit less painful and quite a bit more robust with the Falcon microscope that we've been using from Leica.
Thank you so much. And our next audience member wants to know, how do you train people in these cutting edge tools?
Well, so that's an interesting challenge. And one of the things that's been nice about Enrico Grattan's formalism of using the phaser is I think it's much easier to get an intuitive feel for how the processing is going on, and also be able to interrogate the data in a way. One of our researchers, Francesco Cotrale, talks about it almost as ping-ponging between the phaser display and the real image display, and being able to go around and discover different features in different regions within the tissue. So the training has become a lot more intuitive, and the equipment has gotten much more integrated in being able to do that. Some of our early instrumentation required one computer to run the microscope, another to run the analyses. And those sort of offline or running in parallel approaches have been much more of a challenge, because while they're collecting, they have to imagine what the result might be. Than the integrated tools that we have now with the Mika, for example.
Thank you so much. Now, how do you benchmark or validate the findings?
You know, in imaging, this is one of our big challenges, and one that I think we really face as a field. How do we move from a picture to knowing that that picture is capturing something valid? We have been doing that by trying to collect images with different numbers of labels or different numbers of photons, and looking at the errors that result as we go to minimal dose imaging. It's something I think that's going to be a challenge for all of us, and I think we're going to have to really embrace, is think about, how do we know that the nice picture we're getting is the picture that actually has the meaningful data in it. I think some of these recent tools are making it easier to do that because the answers are more quantitative and easier to interrogate. But I think it's going to remain a challenge for the whole field for a while, especially as we're thinking about reproducibility of the findings and so on.
Thank you so much, Scott. And I want to thank our audience for these great questions that are coming in. Scott, can these tools become routine enough that pipelines can be created?
Well, that is what we hope for. And in fact, that's what we think we're establishing right now for some of our collaborators. By pipelines, I will put words in the person's mouth, but I will turn it into a question I can answer. By a pipeline, what I'm thinking about is some sort of protocol I can give people for the collection, the processing, and even the figure rendering of the data. And together with Carl Kesselman and a number of colleagues within the Translational Imaging Center at USC, we've been trying to build an environment in which the figure legend gives people a DOI that links them to the data collection pipeline, to the raw data, and to the tools. We're hoping that that sort of pipeline will help us in interrogating and re-interrogating the same data, and also in distributing that as a pipeline. If others want to collect similar data, they can look at that digital object identifier and be able to find the tools and the pipeline and all the details that normally you might have to go through 6 or 8 papers to find, because people always refer to another paper that they did some other aspect within. So we're excited about the possibility not only of creating these pipelines, but making them facile for others to use. And we're hoping even to provide some of these through an NIH grant that we're just waiting to hear word on, because we would like to be able to provide this to the field in general, ways to get at answers in some of these molecular imaging questions.
Absolutely. Now, our next audience member asks, is there a theoretical and practical limit to how many spectral contributors can be separated?
Yes, it's a moving standard. Many people think, for example, that if you collect 3 spectral bins, that you can separate 3 labels. And I would argue that's not the case. Once we move into the Fraser space, we can see in our data and Enrico Grattan's and many of the others that have used this sort of approach that small differences can be captured that are very predictive of labels. We can also see cases when the discrimination fails, and cases in which the photons, there are too few photons to make a reliable call. So I think in all of these cases, there's no theoretical limit, but there's a clear practical limit. And what we try to do is drop our photon budget by a few fold and see if we can still make the discrimination. This is why we did the machine learning tools in part, is to see what we could reconstruct from that, but to understand the failure modes. And I think it's going to be incumbent on all of us, not just to blindly assume if I'm collecting 4 channels, I can do 4 labels, or if I'm collecting 10, I can do 10 labels. I would argue that that's not the case. Instead, we have to really validate in our setting where the indeterminacies happen.
Thank you so much.
I also noticed that somebody asked a question about how small the differences can be, and that how small the differences can be very, very subtle. For example, a few years ago, we did an analysis with 5 different green dyes doing a multi-spectral, multi-label separation. And it looked as good as many of the RGB and far red dye separations we had done on other equipment. So I think that there's no hard and fast rules about the number of labels and the number of bins.
Thank you so much. And to add to that question that you were just answering, that same audience member said like 0.5 NS, and just the follow-up question, how many cells?
Okay, on lifetime.
Yeah. Yes. And how many cells or experiments do you usually carry to make sure the differences is indeed there?
Yes. So the difference in lifetime that we can see depends partially on the photon budget. So how many photons do you collect to make sure that you are resolving the lifetime difference? A half of a nanosecond is more than enough to see major changes and with enough photons being collected. That typically slows down the experiment a little bit, although with the Falcon approach, it's been less of a sacrifice. We've had a much better efficiency in collecting enough photons. It's important in lifetime imaging, just as in single cell sequencing or any other sort of thing where we're looking at discrete differences between cells or between voxels, to do the statistics carefully. And one of our challenges is to make sure that our users are collecting enough cells and enough pixels within the cells to really trust the results and run statistical tests to validate it. We've not had a problem, and so far, none of the issues we've looked at, as we've collected more and more data, have we had to revise what we thought in our earlier analysis. So like with anything, I think it's just using good statistics is key.
Thank you so much, Scott. And again, I want to thank our audience. We have time for a few more questions. Scott, when you say phaser inside and spectral phaser inside, does this mean I need to train people on how to use and read the phaser plot to be able to use that system?
You know, I was a little bit worried when I wrote that there. I put it on the slide in part because I used to have a laptop computer that had Intel inside written on it, and I liked that phrase. In the case of my laptop, I didn't need to know how to use a mask aligner and how to do the lithography on a computer chip, or even how to write lower level code that would run it, to be able to exploit that intel that was inside. And one of the things that I've really liked about the phaser approaches and the Mika implementation in particular is you don't have to be able to think in phaser space to be able to use it, any more than I needed to be a material scientist to be able to use an Intel microprocessor. So it's to the user, it's the underlying machine that makes the separations possible. And that separation is seamless and fast enough that you're not even aware it's going on. And in the same way that you don't think about the cycles of the clock in your processor in your laptop. So I didn't mean to put it in like it was some of our earlier equipment that we've designed that to run required two physicists lurking over your shoulders to adjust the mirrors and tweak things while you are collecting data. This is almost the opposite. This microscope sort of runs like the iPhone of the microscope world. And I've just been amazed at the number of users that have been trained by other users in how to use it. Just the same as if I want to know how to use my iPhone, I have to ask my kids or nephews about the features, because without looking at the manual, they can intuitively work through a lot of the details.
Thank you. Now, how are you planning to use spectral or lifetime phaser on SPIM configuration? Film cameras are typically slow and poor in photon budget. On the other hand, spectral cameras are expensive. Do you have some suggestions about this?
Well, that's a great question. And one of the challenges has been that each of these approaches has had a real cost. So the typical hyperspectral camera or hyperspectral detector sacrifices the majority of the light. It hurts your photon budget in order to get the spectral or the lifetime separation. In lifetime, phaser has been dramatically more efficient with the photons than some of the time correlated approaches, some of the photon counting approaches that are typically employed. And it's why Enrico Grattan has been so successful and why we've enjoyed borrowing heavily from his work. In the hyperspectral phaser, Francesco Cotrale has really changed dramatically what we're able to do in hyperspectral detection. And he and his colleagues have recently made a wide field camera that, even taking into account the efficiency of the detector, is approaching 90% efficiency on the detection and the spectral distinction of the labels. This is dramatically different than the situation with many of the hyperspectral cameras that are out there that have enviable performance, but get that performance by throwing away, 95% of the photons. No, I shouldn't say throwing away. The cost of its implementation is the majority of photons, and it can be as high as 95% are lost. So we're very excited and are now working in our light sheet imaging in implementing that design. That design's also very fast and very inexpensive. And so we're hoping that it, or the next generation of tools that are developed by Francesco and his colleagues, will give us the ability to do hyperspectral detection without the conventional cost that I'm sure the questioner was thinking about. And many of the cameras that we in the past have tried to use, or some of the laser technologies we've tried to use, have just involved putting too many photons onto the specimen and perturb the very thing we're hoping to study.
Thank you, Scott. And we have time for two more questions. Our next question: your examples of new technologies indicate that they actually can be democratized, meaning made accessible for a broader range of users. Can you elaborate a bit more on what trend and what potential you observed here?
That's a very good question. So many of the advanced imaging tools have emphasized advanced and not the imaging side enough, and it has sometimes seemed that the equipment has become very challenging to use. One of the things that we've been trying to do within the Imaging Center at USC, and also in our collaboration with microscope companies, is try to make these microscopes so that they can perform at a high level without having to have, as I was jokingly saying earlier, two physicists hanging over your shoulder adjusting things. I think that there are two components to making that democratization work. One is making the instrument itself so that it's easy to operate at near cutting edge performance without an advanced degree in imaging or applied physics or something of that sort. It might be for that extra little bit at the end that you need the specialized training. But what we're trying to do is give performance that's way better than good enough in a setting where there's where the instrumentation is a help rather than a hindrance. The other important thing is having protocols for the use of that equipment that are generalizable, that it's possible within your figure legend to, as I mentioned earlier, put the protocol and be able to almost distribute to others the set of tools and the set of parameters that were used. And I'm very encouraged in our recent publications that the journals have worked with us on that. One of our projects with Carl Kesselman and Don Arnold and others here at USC, we were able to put digital object identifiers in the figure legends that take people to the details of the protocols, so that it's not something where you have to be an expert to interpret what was said or what was implied in making the experiment work. I think the equipment manufacturers have been doing a great job too in making these instruments much more user-friendly rather than performance only oriented. Some of the early scopes were much more like trying to drive in an open go-kart. you can go really fast, but I wouldn't want to carry the family in it. And right now, I think the microscopes are getting to be like very high performance family sedans or something of that sort.
Thank you, Scott. And we have one final question. I'm very interested in Muse. Is that an established as commercial kits would be, or does that involve the miracles of a 10-year trained technician?
That's almost the same question as the last one, but in a very different area. And the questioner is right that many of these technologies have been painful. And recently, HCR reagents are now possible to purchase from a company from Niles Pierce and his colleagues called Molecular Instruments. The Muse reagents are taking a little are more they're newer, so they're more early in their design trajectory and all. But there's a group in Switzerland that's been developing it under the leadership of Simon Restrepo, and he and a startup company there have been working with users, generating kits that others can use, and have had very nice results, and even a couple publications coming out using them. The rapid adoption that I've seen from others of the Muse technology suggests that it doesn't require the 10-year expert, and it doesn't require the double secret protocol that isn't written down to make it work. We've all been through those protocols, and what we're enjoying is finding something that can work more robustly and can be handed to a new user in the lab and get the results. And so far, it looks like Muse is passing that test.
Scott, I want to thank you for your time today and for your important research. And before we go, I definitely want to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. And before we go, I want to thank all of our audience members for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Thank you, Scott, and everyone. Bye-bye.
Thank you. Bye now.